Okay. All right. So it's 9 o'clock sharp. Let's get started. Hello, everyone. Welcome to today's Zoom seminar. Again, I want to still say great thanks to our speaker, Ashley, for deliver us this lecture during the weekend. Thank you so much. And obviously everyone here online to be with us during the weekend. So it's really my great pleasure to welcome Dr. Alicia Moseman from Duke. Alicia is currently assistant professor of integrative immunology in Duke University. So Alicia got his bachelor degree from Carleton College in Northfield in 2002. And then he continued to pursue his PhD in immunology in Harvard in 2011. So he did a great job in Uli von Andrus' lab, and then he identified machinery by which lymph nodes resident subcapsular sinus macrophage are specialized to facilitate both innate and adaptive antiviral response, which I still remember the paper. I think that time we did a journal club on that publication. I was quite a wave. So as a postdoc, Alicia did the training in Dorian McGavin's lab at NINDS NIH. He addressed the long-standing observation which many non-cytopathic virus fail to generate robust neutralizing antibody response, demonstrate that interferon 1 signaling drives CD8 T cell to engage and kill viral infected LCMV glycoprotein, a specific B cell shortly after infection. So currently his lab interested in understanding central nervous system immunity with a specific focus on antimicrobial protection at the olfactory barriers, which we know is a quite unique biological feature on the barrier surface. Unlike the classical barrier surface in the GI, in the respiratory tract, and then in the skin, the olfactory neural epithelial barrier is quite unique in his dictation to neuronal function. His lab actually seeks to define the machineries by which immunological surveillance and then the immune response within the olfactory epithelium to to support both barrier and the neural sensor functions while warding off the neurotropic pathogens. They're using multifunctional intervital imaging to analyze in vivo CNS immune response and then determine how these responses are tailored to the CNS environment. I think Alicia did a really remarkable PhD study and then postdoc training study, and also he continues his success. After he started his independent research program. With these achievements, he received numerous awards, which including the postgraduate award in interferon and cytokine research. And then also he got Whitehead Family Scholar in Duke University from 2018 to 2021. So with further ado, Alicia, thank you so much to be with us today. And then we are very much looking forward to your talk.
All right, thank you very much, Juan. That's a pretty good summary of my career. It's exciting to be here. I know it's a little bit unusual time for me, but it's also great to have the opportunity to share with people who maybe don't have access to, some of our seminar series that we have more locally here. So Juan kind of introduced this idea a little bit, but when I was a postdoc in Dorian McGavran's lab, one of the things that I worked on was understanding how viruses get cleared within the central nervous system, especially cytopathic viruses. And sort of formed the basis for when we started the lab here at Duke was trying to understand why we could even use this system of intranasal infection to infect the central nervous system. And the reason why we do that, for me, it was very handy to just infect mice intranasally and then find virus in their brains. But when you think about it, that seems like a system that really shouldn't work because that seems really dangerous to have anything that's in your airway get into your brain. And so we really wanted to understand what are the systems in place that might prevent that, or how does immunity work in the olfactory surface, which is in the upper airway. And so that's kind of the title of my talk, is just sort of rethinking upper airway immunity, because I think a lot of times people don't really appreciate what that really means. And so actually I'm going to start out with like what I would call just big questions about upper respiratory tract immunity. And that I don't really have answers for all these, but I think it's an important thing for just to take a second to think about it. Because when you say upper respiratory tract, most people think, I don't know what they think. So you can think in your own mind what you think of when you think of upper respiratory tract, and what also do you think that the upper respiratory tract actually does. So you think of breathing in, right? Air passes through the upper respiratory tract to get to the lower respiratory tract, but what does it actually do there? And then sort of questions like, what cell types would you find in the upper respiratory tract? You might think of respiratory epithelial cells, but I've already talked about olfactory senses. So you know there has to be other cells if you're going to be able to smell with your upper respiratory tract. And then another thing to think about is what pathogens infect in the upper respiratory tract, because there are lots of different pathogens that could potentially infect the upper respiratory tract. And what parts of that they infect are sort of specific. Another important question, which I think is actually surprisingly difficult, is how do you actually measure the impact of immunity within the upper respiratory tract? Because a lot of times people will do washes or try to find virus or other microbes that way, but that's not an incredibly efficient way to do it, honestly. And it's actually a pretty tough place to measure the impact. And then, of course, a big question these days is, how do you design vaccines? And SARS-CoV-2 certainly brought this to the forefront for all of us, is understanding vaccine efficacy in the upper respiratory tract. And I think that while SARS-CoV-2 vaccines were extremely efficient at protecting the lower respiratory tract, their efficacy really in the upper respiratory tract, I think, was a little more unclear. But again, the design of the vaccine is to keep people from dying, and for the most part that works, and that's through the lower respiratory tract protection. Okay, so I'll just go to where, let's sort of the lead in here is, like, when I say what is the upper respiratory tract, I'm kind of giving it away a little bit, but I want to emphasize, because we're going to talk about one specific part of the respiratory tract, is that the upper airway is actually not just one thing. It's not just respiratory epithelium that just guides air back into your lungs. There's respiratory epithelium, which, like, if you look at this schematic of a mouse, it's kind of the beginning part of the turbinate structure of your nose. So when the air first comes in, it goes across respiratory epithelium. The same in a human, it comes up through these, through your nose and across the respiratory epithelium. But there's also olfactory epithelium, or you might hear me call it, some places will call it olfactory mucosa. Mucosa would be just like the entire thickness of the thing. Epithelium would be just the surface epithelium of that structure, but we're going to use them kind of interchangeably here. But the olfactory epithelium, the air has to pass across this as well. And what's unique about olfactory epithelium, which you might have guessed, is that it is dedicated to smelling things. It's the olfactory system. So it's not there just to let air pass over it and just make its way to the lungs. It's there really to detect environmental chemicals, odorants around the organism. And so I just want to make sure that everybody understands that that we would call this all upper respiratory tract, but it's very different. The respiratory epithelium doesn't have neurons in it. Olfactory epithelium is filled with neurons. Here you see this image of an olfactory reporter mouse. These are olfactory sensory neurons in green. You can see this epithelium is just filled with neurons. So this is a totally different structure, and it's all in the same area. And really, in fact, we'll see some pictures where they're right next to each other. And here actually is an example of this in a mouse. So this is a mouse's head that's been sectioned sagittally. Again, this is one of these olfactory sensory neuron reporter mice. So all the green you see here are olfactory sensory neurons. You can see it out here, these green neurons are lining the olfactory surface here, and they also extend back into the brain. So you can see these are the termini of these neurons. So one neuron has its cell body out in the airway, and then the rest of it extends back into the brain to actually deliver that smell signal. And so all the area that we would say is covered by green cells here, olfactory sensory neurons, we would call olfactory mucosa, olfactory epithelium. And then the rest of it, where you don't see olfactory epithelium, it's respiratory epithelium. So now you have these two pretty distinct areas, either with and without neurons, that make up the upper airway. And so I already kind of mentioned this, but the one of the really unique parts about this anatomy is that olfactory sensory neurons, in order to get into the brain, they go through this specialized structure at the front of the skull called the cribriform plate. I mean, honestly, I didn't even know this existed until I started working on olfactory infection and these intranasal infections. But the cribriform plate it's the front of the skull, but it has these little holes where the olfactory sensory neurons actually penetrate through the front of the skull and to get into the brain. So what's interesting here is that there's all these barriers to protect the brain, but in this situation, these neurons can move directly from the outside into the brain. So they go from the airway directly to the brain. There's no barrier between them, really. So again, this is a very efficient system for relaying scent information very rapidly, but it also really bypasses all the barriers that the CNS has to protect itself. And so I'll just stop here and say, people often think, okay, what is going on here? I thought I tuned in for an immunology talk, and all I'm hearing about is like olfactory sense and olfaction. That's not really immunology. And I would agree, that's not. And I would say that that's unfortunate, really, is that there's been a lot of work done on olfactory sense, the neurobiology of olfaction, but not as much done on the immunology of the olfactory epithelium and the mucosa, because it is a mucosal barrier. I also want to point out that I mean, I personally love smelling things. That sounds kind of odd, but if imagine your world, there are people who can't smell. If you're one of those, I'm sorry. But the world that we live in is made richer by smells. And in fact, one of the things that we're interested in as a lab is understanding smell loss, both as a function of injury or infection. But individuals who've lost their sense of smell, they really can't experience the world in the same way anymore. And it's a very, sounds trivial, but it's a very depressing situation. Imagine all the foods that you like to eat, all the drinks you like to drink. You basically don't get any enjoyment out of those things anymore because you don't have odors. But I promise this talk is not about olfaction, really just about olfactory barrier function. Because smells are not the only thing that go up your nose. As we kind of talked about already, viruses, other germs can get spewed out into the air, and then you breathe them up through your nose. And once they're in your nose, they can infect the upper respiratory tract. They can make their way down to the lower respiratory tract. But like I also mentioned, that this olfactory surface is kind of like a weak link in the CNS protection, because if you can infect an olfactory sensory neuron, you get inside that sensory neuron, there's really nothing between you and the brain at that point, because you can travel intracellularly from the tip of this neuron all the way back into the brain. And just to show you what this looks like, this is a similar picture to what I showed earlier, but this is no longer an olfactory sensory neuron reporter. This is actually virus. This is a viral reporter. So if you put fluorescent VSV, vesicular stomatitis virus, on the tip of a mouse's nose, they breathe it in, and within 24 hours, this virus is replicated like crazy in olfactory sensory neurons, and then transited across the cribriform plate and into the brain. And you can see this visually here as the green, because it's out in the airway here, and it also crosses into the bulb. But you can also see this with conventional viral titering assays, where if you take out the bulb from these mice, you can see that within 1 day, they can get like to 10 to the 4th replicating virus in their brains. And that's just because the virus was on the tip of their nose and they breathed it in. And so you can see that for neuroinvasive pathogens, like VSV in this case, viruses that can get into structures and invade directly into the brain, this is a barrier tissue that's no joke. You really got to lock this down, because if things can get into your neurons and then get into your brain, it's a much worse phenotype than, if you just get a runny nose or something like that. But I would also say that appreciating the olfactory surface as a surface for replication of viruses is not just relevant for neuroinvasion, because as we saw in SARS-CoV-2, which actually does replicate in the olfactory tissue, it doesn't replicate in olfactory sensory neurons, but it does replicate in other cell types there. But if you don't protect this surface, you provide a substrate for viral replication, even for viruses that don't want to invade the CNS. And so again, you have a hard time sterilizing a population or of sterilizing immunity across the population if you can't protect this surface. So people are always going to be getting infected on their olfactory surface, even with just respiratory viruses like SARS-CoV-2. So we got into this project thinking about like what actually would be immunity to the olfactory mucosa really look like. It's mucosal protection. We know that's important because we want to reduce infection in general. We don't want transmission of respiratory viruses. I mentioned that not all of these are going to be olfactotropic. They're going to be less neuroinvasive, but they can, they'll be less neuroinvasive, but they can still replicate in the olfactory tissue. And so we definitely want to block. What's going on here? Okay. So we want to know, how do we defend this? Do we need some kind of specialized mucosal response, or is there a special olfactory specific response? A lot of people think about mucosal responses and they think of secretory IgA, this isotype of antibody that is really abundant, especially in the gut, other mucosal surfaces. And we didn't really know if that was going to be specifically relevant for the olfactory surface or not. So when we started this project, we really wanted to rely on vesicular stomatitis virus because it's a great model virus, because it does a couple of great things for us as a model. One is we just saw it infects the olfactory route really, really well. And therefore, it's easy to get high titer, it's easy positive readout. It also invades the brain, which is great. We can check 2 different tissues. But even better is we can immunize, or we can infect in different routes. So if we infect subcutaneously or intraperitoneally, you can also get viral replication. And in all of these routes, you get a really strong neutralizing antibody response, which again is useful for us because it gives us almost a binary readout. When we have antibody, if we have neutralizing antibody, you don't get viral replication. And so we wanted to know, if we have antibodies to these things, is it going to protect the olfactory surface? And so to do this, a grad student in the lab, Sebastian Welford, he's getting ready to finish up here, but he was brave enough during SARS-CoV-2 to we had an interesting observation, and we did a detour here. He had one project as his thesis project, and then we're like, you know what, that's cool, we can kind of work on that, but this seems a lot more interesting. So let's kind of shift gears, and he just did that. And I think it was worth it, let's say. So we started out with this really simple question, which is like basic immunology. And that is, if you infect a mouse, can you reinfect that mouse intranasally? Like, is the prior infection going to protect intranasally? And so here's the setup. We have a primary infection either intranasally or intraperitoneally. And then we come back and we challenge these mice with the same serotype of virus intranasally. So we're now testing olfactory protection. Intranasal is essentially just going to read out, do we get replication of virus in the olfactory tissue? And you can see if you don't have the primary infection, you just challenge a naive mouse, you get a lot of virus in the olfactory tissue after 2 days. This is what we expect, like a ton of virus replication here. But you can see that in both situations, intranasal or intraperitoneal, no replication at all. So this is exactly what you would expect. This is like what we would call basic memory response of infection. Like, antibody has protected these mice. And we know it is antibody because we actually have 2 different flavors, let's say serotypes of VSV, one Indiana and one is New Jersey. And the New Jersey doesn't have the same B cell epitope. So the neutralizing antibodies to Indiana don't neutralize New Jersey. But they do have they have conserved T cell epitopes. So the T cell response to Indiana should be able to protect against New Jersey. But what we see is if we challenge first with New Jersey, and so it has T cells, but it doesn't have antibody, and you come back and challenge with VSV Indiana, you see that you really don't protect the mice at all. So this indicates that any protection that you're getting here through the re-challenge is due to the antibody response, not the T cells, because we have T cells against VSV here, but they're not protecting at all. So now we know we can really focus on the humoral response here. And we've really taken away almost nothing from this experiment. It's pretty obvious is that if you infect something, you can't infect it again. Okay, it's like really basic immunology. And that this olfactory surface protection, actually preventing entry of the virus into the olfactory mucosa, is antibody dependent. All right, so the next question we asked was a little bit more interesting. And that is that because we had assumed that when we measure serum antibody, which is a pretty conventional way to check whether an antibody response has happened, is that we would think that this was protective. So if you have a primary infection, you have a serum antibody, you check it, like, okay, we have antibody, then we should be protected. But to test that, if that's true, what we did was we injected antibody, either hyperimmune serum from a previously infected mouse. We transferred the serum into a naive mouse, or we also have a monoclonal antibody that's neutralizing against VSV. So we can transfer either monoclonal antibody or convalescent plasma that has a lot of neutralizing antibody already into these mice the day before we infect them, and then we infect them, and then we look 2 days later to see what impact there was on replication. And what you can see, actually, somewhat remarkably, is we were able to, initially, we can see that we are able to achieve a neutralizing antibody titer in circulation. That's essentially the same between an infected mouse, here IP infected, and a mouse that's received convalescent plasma. So in our monoclonal antibody. So we've able to transfer enough antibody to sort of replicate what the antibody amount a regular infected mouse would have in the memory phase. But what we see is that this is actually olfactory mucosa, but the same is true in the bulb. But you can see that this antibody does not protect against replication in the olfactory tissue. So whereas naive mouse is very high replication, once you have an infected mouse, it's not protected. But if you transfer neutralizing antibody to those animals, they're still not protected. So these mice have a lot of circulating and neutralizing antibody, but yet they're not getting protected at the olfactory surface, which was really surprising to us because we assumed that these would be interchangeable. If you have circulating antibody, you would be protected. But this is clearly not the case because these animals are replicating virus almost like they're naive, but they have lots of antibody in circulation. These labels got shifted. I'm sorry, guys, but this is actually the olfactory bulb titer. So this is in the CNS, and you can see that in these individual mice where they aren't protected in the olfactory mucosa, they are also not protected in the CNS. So they still got virus in their brains as well. And interestingly, if you look on the lymph nodes, so if we were to challenge these same animals, not the exact same animals, but the same conditions in the foot, like to a subcutaneous infection, we can see that the amount of antibody that they have is able to protect them against a peripheral infection. It's just not able to protect them against an olfactory infection. So this was really surprising to us because we had, again, assumed that serum antibody was going to be protective. But then we wondered, okay, we only transferred antibody overnight and only let it sort of soak into the system for 18 hours. Maybe that's just not long enough. Maybe for some reason, movement of antibody across these tissues takes longer than we would have expected it to take. So to get around this, what we did was we did use a parabiotic approach where we took a naive mouse and then we surgically attached it to an immune mouse. So this mouse was infected with VSV ahead of time before we attach these guys. So this mouse, though, his blood is full of neutralizing antibody. And once these mice are attached, they're going to share blood, and this animal is going to provide antibody to the naive mouse. And that's actually what happened. So these are the partners. They have different neutralizing antibody titers, but their naive partner has the same titer that they do. So they have fully mixed their blood and their serum antibodies together over, let's say, 2 to 3 weeks. And then when we challenge these mice, what we see is that the immune partner is protected, where the naive partner is really not protected from this challenge. Again, suggesting that just because you have serum antibody, it doesn't result in protection. And we found this, again, to be pretty striking. For some reason, antibody is not able to move from one immune partner into the other partner in a way that's actually protective at the olfactory surface. And just as a sort of, these were obviously striking results, and we were like pretty excited, but we also wanted to check to see if this was something that was only specific to VSV, something weird about the VSV response. And so to do that, we used another infectious system, which was influenza B, Malaysia 04, which we had coincidentally identified through another collaboration we have with Nick Heaton here at Duke as a type of flu that will infect olfactory sensory neurons. Some types of flu do, and some types of flu do not, but this one does. And what's nice about it actually is that it infects both respiratory and olfactory epitheliums. And so that gives us another way I'll show you in a second, to compare. But just as an example here, if you transfer convalescent plasma against MAL04 into a naive mouse and then infect it, you can see that, again, the convalescent plasma does not protect against olfactory replication in these animals. compared to intranasal infection. So again, this looks like what we saw with VSV, where the antibody is able to get into circulation. In fact, we have even more antibody. We were able to transfer a lot of antibody to these mice, and it was not protective at the olfactory surface. So this is not specific to flu. And this is kind of gets back to this idea of respiratory versus olfactory. What's kind of neat here is you can see that when you transfer convalescent plasma into these mice and then infect with Malaysia 04, you can see the convalescent plasma doesn't affect the infection here you see as green, Malaysia 04. Convalescent plasma doesn't protect against infection in the olfactory tissue, but if you look in the neighboring areas of respiratory tissue, it is protected. So you're actually able to specifically protect against respiratory replication with serum antibody, but you don't impact olfactory replication. And this is reflected here when we quantified these neighboring regions of respiratory versus olfactory replication. You can see that the convalescent plasma respiratory epithelium was protected, whereas convalescent plasma olfactory replication is not reduced. So what the heck is going on here? We want to know, why is this not working? We had our suspicions, but we had to have a way to really see this. And so the approach that we used was to inject antibody that would bind to EpCAM, so epithelial cell adhesion molecule, I believe. And that's expressed throughout the olfactory and respiratory tract. So you can see here, if you stain in white is the EpCAM staining. If you just take a tissue section and stain it, you can see that there's white staining throughout the olfactory tissue and also the respiratory tissue. Everybody is staining with EpCAM here when you just stain it on a slide. But if you take that same EpCAM antibody, come on, buddy, and you inject it intraperitoneally into the mice and then let it leak out into tissues where it has access to bind to its epitopes, what you see is that the EpCAM actually binds like crazy out here in the respiratory tissue where you don't see olfactory sensory neurons, again in green. And the olfactory sensory neurons, all this olfactory tissue out here doesn't pick up antibody. And in fact, if you look really carefully, like if you zoom in on these, you'll see where the respiratory tissue is, where there aren't olfactory sensory neurons in green. They have staining here for EpCAM, but as soon as you start to see neurons here, you lose this EpCAM staining. So this to us was a sort of visually striking representation of the fact that antibody doesn't seem to be able to leave the blood and bind to these olfactory tissues. It can leak into respiratory tissues where it binds to its epitope, but it's not able to actually get into the olfactory tissue. And that would, of course, explain why we don't get binding. We don't get protection in these protection from serum antibody. And we've kind of coined this the blood olfactory barrier, the BOB, because it seems to form an endothelial level barrier that blocks serum antibody or probably anything of large molecular weight from actually leaking out of circulation into the olfactory mucosa. What's going on? Okay, so we really wanted to understand. We knew that prior infection was protecting these things. We knew the antibody was important, but just to kind of cover our bases, we went ahead and depleted CD8 T cells or NK cells that didn't have any effect on the protection. But we knew also that IgA has this long history of important roles in mucosal immunity. So we wanted to test whether IgA was maybe why this wasn't working. So maybe if you infect a mouse, it generates protective IgA that for some reason isn't crossing in the same way between partners or when we do serum transfers. But actually, when we infected mice that were IgA deficient, it didn't make any difference. Those mice were still protected just fine. So we don't think that this is dependent upon IgA or a specific isotype of antibody. But what we think it is it's requiring the BOB, this barrier that prevents serum antibody from coming in, now requires that you actually have to have antibody producing cells, plasma cells, in the tissues themselves, the olfactory tissues, where they can produce antibody on the other side, the tissue side of the blood olfactory barrier. And then when that happens, the antibody is provided out to the surface of the olfactory tissue where it can be protected. And we actually can see this. So if you quantify the number of plasma cells, you can see there are more plasma cells in the tissue after you infect the mice. But more specifically, what really is the best way to do this, because these cells are relatively rare, is we used an ELISpot approach where we took the olfactory mucosa. We actually have to strip it off of the olfactory turbinates because the turbinates are full of bone marrow and that kind of complicates things. But if you take that whole mount, pull the mucosa off, and then you digest it, and then you plate it, you can actually detect viral specific antibody secreting cells in the mucosa of infected mice, whereas you can't detect that in naive mice, of course. So this really strongly suggests that what we have here is we have antibody secretion locally, not from the blood side, but actually in the tissues that's serving to protect previously infected mice. And then to test this really more formally, we use an approach to deplete plasma cells. Now, not specifically olfactory plasma cells, but all plasma cells, but we think that this is still an interpretable result. So what we do is we infect the mice, and then we come back 3 weeks later, and we either treat with this proteasome inhibitor, bortezomib, which, because plasma cells have such a high protein synthesis, when you inhibit their proteasome, they really don't like that, and they tend to undergo apoptosis. So this is used actually clinically to treat multiple myeloma, and it will kill plasma cells, both wherever they are. And the other approach we use, which I'll just go ahead and describe right now, is we use an AID-CRE crossed to an inducible diphtheria toxin receptor. So this is not selective to plasma cells per se, but it is going to kill basically all class-switched or hypermutated B cells. And that includes the plasma cell compartment. And so when we do this, what happens is when we deplete with bortezomib, you can see that this reverses the olfactory mucosa protection. And you can think, okay, maybe that's because you depleted all plasma cells and there's no antibody, but that's not the case, because serum Ig has a pretty long half-life. And you can see that the serum antibody is actually not really decreased at this point. So if you deplete only for like 14 days, you give it enough time for antibodies being produced locally to kind of dissipate out of the tissues, but the antibody that's in circulation actually is still around enough that it still has a neutralizing antibody titer. So these mice, even though they still have circulating antibody, we've now gotten rid of their ability to be protected. So this again suggests that plasma cells are needed to be continuously producing antibody in the tissues, and they can't rely on circulating antibody to do this, to protection. And again, the same thing is what happened when we do the AID approach. Again, we deplete all these antigen experienced B cells, and the protective ability is lost here, even though you still have circulating antibody. So as a sort of intermediary summary, we'll just say that you need to have pre-existing antibody if you want to prevent infection, especially neuroinvasion with VSV, because these viruses, they're coming in and they're going to attach to the surface of the cell immediately and enter that cell, and at which point they're no longer going to be available to antibody. So you have to have antibody being produced all the time to meet those pathogens when they come in. So you got to have pre-existing antibody. Serum antibody is not what you need, though. So serum antibody actually cannot reach this olfactory surface. So you got to have plasma cells located in the tissues in order to protect against this at the olfactory surface. But that leaves a lot of questions, and we were still working on many of these questions, but I'll go into a couple of these things that are sort of like the first questions you ask. And one of those is, what role does CD4 cells play? And are all isotypes of antibody protective? I already showed you that you don't need to have IgA, but that's not all isotypes. And then also, we still don't quite understand, but I'll talk a little bit about why plasma cells go into olfactory tissue, because I didn't really emphasize this, but this is an infection that we're doing intraperitoneally. It's not actually at the site of where we're testing in the olfactory tissue. So there's something about a systemic infection anywhere, that is able to drive cells into olfactory tissue, which we thought was pretty surprising and interesting. And is this protection, do you get that no matter what happens? Like, does it have to be a certain type of antibody response to be olfactory protection, or some special sauce, so to speak, of endogenous humoral response? Okay, so we'll just skip to the CD4 T cell requirement. So we all know, or should know, that CD4 T cells are really important for most humoral responses. Without CD4 T cells, you don't get good antibody responses. This goes for both some extrafollicular, but certainly for GC-dependent antibody responses. And you can see that if you deplete CD4 T cells before you infect these mice, it totally abrogates protection, which I guess is pretty much what you would expect. Like, you need to have CD4 T cells to help. This olfactory protection is not driven by, T cell independent process. This is dependent. And sort of interestingly, when we came in and we blocked CD40 ligand, which is an important co-stimulatory molecule that T cells provide to B cells as part of their T cell helping function, this also, even after day 3, at day 3, this was able to interrupt the protection that we saw in in the olfactory surface. So we would interpret this to mean that there's T cell help going on at least after day 3 that is protective here. Even though in this case, these mice generated a good amount of antibody, the mice that don't have CD40 ligand blockade, there's something else that's going on here after day 3 that's actually protecting, leading to protection in the olfactory surface. You'll see this again here. I just want to point out that we also use this beta-mercaptoethanol approach to dissociate IgM. So the IgM's neutralizing power is decreased to basically none. And this allows us to see how much IgG is being produced, focusing specifically on the IgG, protective neutralizing IgG instead of all Ig classes. And you can see here when you deplete CD4s, you really don't get class switch, which is expected. And if you can get some class switch if you deplete CD4 or block CD40 ligand after day 3. But when we also want to look at what kind of class switch was needed, because it seemed like you needed CD4 T cells, and that may be for class switch or for something else. And to do this, we used mice that were deficient in AID, which is required for class switch and hypermutation of the B cell compartment. And what we saw was that if you infect AID deficient mice, they're not protected upon rechallenge, which we thought was kind of interesting. But the clear implication of this is that you require class switch. These antibodies are neutralizing. It's not that they're not neutralizing. They have affinity that's sufficient. And we interpret this to mean that IgM, which is like the native class before class switch, is probably too big for some reason. It's this large pentameric structure, too big for some reason to move through the olfactory tissue in a way or something to be protective. So we know that you need class switch. You can't rely on IgM to do this. And that's probably one of the functions that CD4 T cells are important for, is you actually have to get class switch. But it's not just that, it's something else. Oh yeah, and as I mentioned, without AID, you don't have any neutralizing IgG. Everything you have here is IgM, because as soon as you use beta-mercaptoethanol, this neutralizing power is lost. Okay, I still got time. So one thing we wanted to know is like how these plasma cells actually get to the tissues. And one of the first things I thought of was CXCR3, which is a chemokine receptor that's used all the time by many different cell types to get into inflamed tissues. So this is an inflammatory chemokine receptor, comes up on a lot of effector things because its chemokine ligands are expressed, or type 1 interferon dependent, interferon gamma dependent. These are things that come up when there is inflammation, especially viral inflammation in tissues, and it helps draw effector cells into those tissues. And so I thought, okay, let's look at CXCR3 deficiency. And when you see, we had 2 different approaches. One was using a knockout for CXCR3, or we actually just blocked CXCR3 with antibodies. And in both cases, if you have a mouse that's deficient in CXCR3, you impair olfactory protection, which was actually kind of expected. We thought, okay, maybe they're using CXCR3 to get into the olfactory tissue where they are protected. And similarly, if you use an antibody-based blockade, again, it impairs protection here, but it actually doesn't have any impact on the neutralizing antibody titer. But again, if you remember, this was a peripheral infection. The weird thing here was we thought, all right, but that doesn't really make sense. If we're infecting peripherally, why would there ever be chemokines recruiting cells in the olfactory tissue? Because there's really no inflammation in the olfactory tissue. We've looked really hard to try to find evidence that maybe virus got into the olfactory tissues. Even when we do peripheral infection, we never see any evidence for virus or actually for these chemokine ligands. So we know that CXCR3 is involved, but doesn't seem to be involved actually at the olfactory surface, which was, again, quite surprising and not the easiest way to interpret this. But then what we thought was, oh, maybe this is something that's happening in the lymph nodes, not in the olfactory tissue. Here actually are the CXCR3 ligands in the olfactory tissue. You can see for CXCL9 and CXCL10, these are the 2 ligands in black 6 mice. And they really don't have any upregulation in the olfactory tissue, which is what we expect because we don't think virus gets there. But when we look in the lymph nodes, we can see that there's actually the presence of both of these ligands. They're upregulated in the draining lymph nodes of these animals. And so we don't really yet understand this. We have some bone marrow chimeric data that suggests you need CXCR3 on both the B and the T cell side. And we think that maybe what's going on is CXCR3 is programming an interaction between these 2 cell types in the lymph nodes that is somehow important for providing whatever signal it may be that then drives the cells out of the lymph nodes and into the circulation where they can get into olfactory tissues. But it doesn't seem like this is actually acting at the olfactory tissue. And then the next really big question for us was trying to understand whether vaccines could do this, right? We were concerned, this is like during SARS-CoV-2, do the vaccines that we're getting, are they actually generating serum antibody titers and olfactory protecting plasma cells, or is there some kind of special adjuvant you would need to do this? And the way we tried to do this, we actually tried many, many different types of adjuvants, but we used VSV that we had inactivated with UV light, so it doesn't replicate anymore. So it's essentially just a protein antigen. And we give it we immunize mice with either just VSV alone or VSV mixed together with alum, sort of conventional adjuvant, or with CPG. We also tried other adjuvants. If you want to, you can look at the paper, but I can just tell you that they all work kind of the same for the most part. They don't really work. They do work to give you an increase in serum neutralizing antibody titers. So they work as adjuvants, which is great. So that part works. We actually can increase the amount of antibody that we get by adding adjuvant. Again, a classical concept. And this serum antibody is able to protect against a subcutaneous infection. So this neutralizing antibody, as we expected, did protect against peripheral infection. But sort of to our regret, I guess, is that this is not protective at the olfactory surface. So much like a serum antibody transfer, we have a lot of serum neutralizing antibody, but that antibody alone is not able to protect against olfactory infection. And this again implies that vaccination gives you the kind of antibody response you need to generate a serum antibody level, but not enough not the right kind of response to drive plasma cells into olfactory tissues where they are protective against an intranasal infection. Well, we tried a lot of different adjuvants, but one adjuvant that we actually sort of stumbled upon this while reading some literature. I had never heard of this before, but it's called DMLT. It stands for, I think, double mutant of LT. LT is a labile enterotoxin, and enterotoxin has been used as a mucosal adjuvant before. They ended up mutating it because it's pretty, as its name implies, pretty toxic. It makes people sick. But this mutated version apparently does not do that. But it still has many of the adjuvant properties that the enterotoxin had. And when we use this DMLT adjuvant with a single immunization, like we would with an infection, and then we came back and challenged those mice, we saw that that's not protective as a single immunization platform. Both when we treat the mice IP, intramuscularly or intranasally with DMLT, it doesn't matter. But when we come back and boost those animals, what we found actually was quite surprising. We still don't quite understand what the factors are, but this DMLT given intraperitoneal twice was able to actually generate protective olfactory response, which is really interesting because obviously with infection, you only need one infection and that's enough to give you olfactory protection. But this prime boost requirement with DMLT suggests that there's something that you don't get with the single immunization that the second boost really now allows to happen that's protective in the olfactory surface. I also just want to point out that the intranasal immunization is really challenging. It doesn't work very well, probably for a lot of reasons, but one of those we suspect is because antigen is just not held in the intranasal tissue very long. So you don't really get much response at all to intranasal vaccination, regardless of like what adjuvant we try to use. There are obviously some ways to do that, but it just doesn't work very well. Intramuscular worked a little bit, but not like the IP did. Okay, so I would just summarize what I'm just talking about, and we'll go over a little bit of the what I think the big implications are, but the take-home messages here are that antibodies can be completely protective and really protect the olfactory surface from infection if you have them being produced locally. And that's because circulating antibodies can't effectively get into the olfactory mucosa because there's this blood endothelial level barrier, the BOB, that really prevents large molecular weight things from getting across and into the olfactory tissue. And so you can't rely on bleeding a mouse or say a human even. You can't relying on the serum titer to tell you about what the protective power will be at the tissues necessarily. Maybe in the respiratory tissue, but not in the olfactory tissue. And that the olfactory tissue is going to really rely on these tissue-derived antibodies because, again, they don't have access to what's in circulation. They really need to have local production of antibodies. It's very similar to the brain, where the brain doesn't have access to the antibodies in circulation because the blood-brain barrier prevents those large molecular weight things from coming in. But just unlike the brain, the olfactory tissue is actually out in the airway. The brain is at least a little bit protected. And unlike infection, this is also really, I think, an important finding is that not all vaccines are going to produce this protective immunity at the olfactory surface. So you can get vaccines that do a great job of generating neutralizing antibodies, but they're not necessarily going to give you protection at the olfactory surface. You can increase the circulating titer really, really high, but if you don't have cells that go into the tissue, it's not going to be protective when you get that neuroinvasive challenge. And so again, for olfactory pathogens, these data strongly suggest that the serum antibody titer is not predictive of protection. You just can't use that to know whether you're going to get protection or not. And so I was thinking about this, and this is, I think, something I want, if you can take away anything from this, just think about the upper respiratory tract as more than one thing. It is not a monolith. And in doing so, if you ever want to work on infections or protection of something in the upper respiratory tract, you got to consider them. You got to consider the anatomy first and foremost. And so I just want to highlight a hypothetical situation here where you want to do a vaccination or protect against infections with different pathogens. And I already told you that there are pathogens, some types of flu, for instance, some pathogens only infect the respiratory mucosa. There are some that infect respiratory mucosa and olfactory mucosa, and others like VSV that really only infect the olfactory mucosa. And if you don't know that, let's say you're flying blind, you do not know the pathogen tropism, and you're testing a bunch of different vaccination strategies, and then you challenge with your whatever pathogen it is. Imagine all sorts of different ways you're doing this, and then you challenge those mice, and then you read it out with, maybe you take the whole upper respiratory tract out, or maybe you take out just washes or tissues or whatever, and you check, and you see one of them doesn't have any replication. So yeah, we got complete protection. And this one did have virus, so that doesn't protect. When this one does have virus, so it doesn't protect. Is that what it means? Is that really what that means when you see virus or no virus? Because I've already told you that depends on which tissue you're actually infecting and then what tissue you're protecting, whether you get replication. So you could think hypothetically, if your vaccination, whichever one of these many flavors of vaccine you try to use, if it only drives a serum antibody response, and if your pathogen is only tropic to the respiratory mucosa, that's going to work great. You're going to see no replication in the respiratory area, and you're going to think, I did it. I made a protective vaccine. This platform is great. Or maybe this adjuvant is perfect. It's going to protect the upper respiratory tract against all pathogens. But if you have another pathogen that infects both respiratory and olfactory together, and you assume that you have failed to protect here, but you've only failed to protect the olfactory mucosa in this situation because the serum antibody titer is going to protect the respiratory mucosa, but not the olfactory. And the worst case scenario is that your pathogen is tropic to the olfactory mucosa, and you don't necessarily know that. You achieve serum titer, and then you test again, and you see that there's no change at all in the protection. You don't really know what's going on here. The other possibility is that you don't protect any of these, and so really you failed, but you don't even know that. All you see is virus. And so I guess what I want people to realize is that if you don't know where your pathogen of interest is replicating, you really can't interpret whether your vaccine works at all. I mean, you can test for serum antibody, but you can't read out what these the protective capacity is because you don't know which of these compartments you are trying to protect. So it might actually work in some ways. It just you won't be able to appreciate that. Yeah, so this becomes a nightmare scenario when you're testing different pathogens or maybe different pathogens and different adjuvants, and the different pathogens have different tropism. So you're really you're comparing apples to oranges, as we would say. You cannot compare these things because you're trying to protect different things. You're not really just trying to protect the upper respiratory tract. You're trying to protect either respiratory mucosa or olfactory mucosa or both. So again, if there's one thing I want you to take away from this, is that if you're testing for upper respiratory tract protection, you really have to know what are you interested in knowing the protection of, the respiratory mucosa, olfactory mucosa, or both. And that might take some work. You might have to look to figure out where you're actually getting antigen generated, because it could also be vaccinations, immunizations, not just infections. All right, so these are the big take homes. You really got to distinguish upper respiratory tract issues, got to know the pathogen tropism. We want to design vaccines that impart olfactory immunity. And that means we really have to know what, we need to know what are the special sauces, the special signals that cells get to do that. But also we need to know where the pathogen infects in order to know if we've protected or not. And, you know, we actually did, I'm not going to show that data, I guess, but we actually tried to use these Pfizer BioNTech, like early during SARS, we managed to get some SARS-CoV-2 epidemic. We got some leftover vaccine vials from Duke immunization clinics, and we tested these in mice to see if they would generate olfactory protection. And they did generate sort of pretty decent olfactory protection. But I think that in the real world scenario, what we've seen is that vaccination against SARS-CoV-2 does a great job of preventing severe pulmonary infection, but doesn't necessarily stop the upper respiratory tract infection as well. I'm a real world example of this. I got infected by SARS after I've been vaccinated many times, SARS-CoV-2, and I had almost no lung phenotype at all, but I had a crushing upper respiratory tract infection. And I always assume this is my Bob's fault for keeping the antibodies out, but we don't have evidence for that, but it's a strong anecdotal link, let's say. All right. And so I'll just finish up. I want to thank the people in the lab who helped do this. I already mentioned Sebastian. He's a very capable guy, but you also have to be brave. You're a thesis student just to switch directions and try something else. Annie in the lab is very helpful with a lot of different things. And Kiana was helpful also with some technical issues, a technician in the lab. And everybody else is obviously supportive. We had a lot of help for different things. Garnet Kelso's group was very helpful of antibody stuff, and Nick Keaton's group helped us with the getting started doing Malo4, which we kind of continue doing. And then of course our funding agencies. I should also thank Dorian because he, let me take these olfactory things and run with it, and got me started down this track with olfactory and CNS infections. And I probably wouldn't be here, I definitely wouldn't be here right now without his support. And with that, I guess I'll take questions if we have time.